We now welcome Dr. Praveen Cherukupalli to lead the virtual session on risk based approaches to impurity management, control strategies in final dosage form.
Good morning, everyone. So thanks for the EBM group and especially Ms. Guneet for organizing this Pharma Impurity Conclave event. Okay, so many times we had personal events. So this time, okay, so EBM has organized virtual event. Good to see all. Just I will discuss uh, risk-based approaches to impurity management, control strategies in drug substances and drug products. So, risk-based impurity management in drug substances and drug products involves a systematic approach to identify, assess, and control impurities that could pose potential risk to patient. So this approach focuses on minimizing the presence of harmful impurities while maximizing the safety and efficacy of the final product. Okay, for having controls in impurities in drug substances or drug product, risk-based impurity management is very critical. Yeah, what are the benefits? Okay, so by doing a risk-based impurity management, so first thing is improved product quality and safety. So by controlling impurities, the manufacturer can ensure that the final drug product is safe and effective for patients. And reduced regulatory scrutiny. Demonstrating a commitment to risk-based impurity management can help to streamline the regulatory review process. So during the risk assessment, okay, so you will give complete control strategy for all the impurities, all the possible impurities, process-related impurities, degradation impurities, and so effectively demonstrates about the impurities. So this will minimize the regulatory scrutiny and ease your regulatory approvals. Next, enhanced manufacturing efficiency. Process optimization and other control strategies can lead to more efficient manufacturing process. So when you are having complete idea on the impurities and impurity profiles, definitely you will have a good process where a manufacturer can efficiently manufactures the drug substance or the drug product. Next, cost savings. By preventing product recalls or regulatory issues, manufacturer can save a significant cost. See, suppose the recalls, whatever the recalls we'll see, that is only because of the inadequate knowledge on some of the things, or in rush in releasing the product into the market. So when you are having appropriate control strategy for all the impurities, so you can obviously you can avoid the recalls. By that you can cut the costs of the company. Yeah, just so what is the impurity? Okay, so in the drug substance, any component in the drug substance that is not the active ingredient, like that, not the chemical entity is defined as a impurity. In the drug product, it is the APA and excipient. Other than the APA and excipient is called the impurity. Now, so why we have to remove the impurities from the drug substances or drug product? What if the impurities are present in your drug substances or drug product? Okay, so obviously the impurities will reduce the drug efficiency. Impurities can compete with the active pharmaceutical ingredient for binding sites on target molecules, thereby reducing the drug's therapeutic effect. Although they are present at very minimal level, the impurities compete in the binding sites of the target molecule. So obviously the efficacy will be of the drug will be reduced. Increased side effects. Some impurities may have inherent toxicity or unintended biological activity leading to adverse reaction in patients. Like thalidomide case, where the enantiomer, other enantiomer of the drug so is highly dangerous. So because of that, so it will give a teratogenic impact on the patients. So unknowingly, having the impurity in the drug substances makes the drug as a very toxic and decreased stability. Impurities can act as a catalyst for degradation, causing the API to break down over time and lose potency. This can shorten the shelf life of the drug and render it ineffective. So, when impurities, some of the, in not all the cases, some of the impurities may act as a catalyst and will deteriorate your API. And because of that, okay, the API efficacy will be reduced. Incompatibility issues. Interactions between impurities and other formulation components can lead to physical or chemical instability, affecting the drug's delivery or effectiveness. So all these, okay, so either the efficacy may be reduced, it may be having negative effects or decreased stability and incompatibility issues. Many, because of these reasons, the impurities, okay, so should be minimized in the drug substance or drug product. And what are the types of impurities which will be present in a drug substance or drug product? So, as you know, okay, so everybody knows there are organic impurities, inorganic impurities, and the residual solvents. So, the organic impurities may arise from the starting materials during the process, uh, like process impurities and degradation products, intermediates, reagents, ligands, catalysts, and if you come into drug product, it is a drug substances excipient interaction and container drug substance interaction. So all these possibilities will be there for the organic impurities. So we have to effectively control these organic impurities as per the ICH guidelines. Now, inorganic impurities, if you'll see, there are heavy metals, any residual metals, reagents, ligands, catalysts, inorganic salts, even charcoal, filtrates. All these comes under inorganic impurities which need to be controlled adequately. And the residual solvents may be volatile or non-volatile. So these are, as you people know, so these are class 1, class 2, class 3 solvents. So these need to be controlled adequately, where we will avoid most of the cases. Okay, we will avoid the usage of class 1 solvents, and class 2 solvents will minimize, and class 3 solvents generally the process chemists will use for the manufacturing of the APIs. So these are the major main guidelines for controlling of the impurities, where ICH Q3A will define impurities in drug substances, and ICH Q3B which gives the details of the impurities in drug products, and ICH Q3 is a guideline for residual solvents, and Q3D it is for the elemental impurities, and Q6A is for the specification in drug substance or drug product, and ICH M7 is for the assessment of mutagenic impurities, including the nitrosamines, and ICH Q9 is quality risk management, where all the risk approaches you can made as per the guidelines of the Q9. This is the impurities limit as per the QC ICH Q3A guideline. So when the dosage is less than 2 grams, the reporting threshold should be 0.05. Identification should be 0.10 or 1 mg. and qualification should be 0.15 or 1 mg. See, if whether uh, the drug is like less than 2 grams, the unknown impurity limit should be 0.10 or 1 mg, whichever is lower. Okay, we can't say that always the unknown impurity limit is 0.10. So suppose this rule, whatever 0.10 rule is applicable up to 650 mg only. When the drug limit is more than 650 mg, obviously the unknown impurity content will be reduced, as the maximum allowed impurity limit will be 1 milligram per day. Suppose if you will take 800 mg API, so if you'll keep unknown impurity as a 0.10, then the total impurity intake will be 0.8 mg. And if you known impurity is 0.15, and 0.15 into 800 mg, it will be more than 1 mg. So because of that, you have to reduce the known impurity content in the drug substances. So whatever the dosage, but the acceptable intake should be 1 mg, whether it is known or unknown. These are the ICH Q3B thresholds. Reporting thresholds for less than 1 gram, it should be 0.1%, and more than 1 gram, 0.05%. And okay, identification qualification threshold is defined as per like a Q3B guideline. And here, one point, degradation products that are also significant metabolites present in animal and human studies are generally considered qualified. See, these impurities, the degraded impurities or metabolites. So we have to make known and put the appropriate limits as per the ICH guidelines. Next, okay, so we have seen the impurities, organic impurities limit in drug substances and drug products, that is ICH, as per ICH Q3A and ICH Q3B. Now, the residual solvents limits shall be controlled as per ICH Q3. So, where class 1 solvents to be avoided, class 2 solvents to be limited, and as because it's limited in the sense the content or the PDEs of these solvents are very less. So obviously we have to control, or we have to use these solvents in limited. And in class 3 solvents, they are having a low toxic potential. We can use these solvents and control as per the ICH Q3 residual solvents limit. Now, if we'll come to inorganic impurities, this need to be controlled as per ICH Q3D guidelines. So all the pharmaceutical industries are doing the risk assessment. Okay, so what is the source for any elemental impurity from the drug substance, like all the process, and for drug product, like what are the ingredients which are used for the drug product preparation. We'll use the risk assessment, and we'll check the class 1, class 2A elements mandatorily, and class 2B, class 3 elements as per the requirement and controls. So we'll use ICP-MS and ICP-OES to check the content of the metals in our drug substance or drug product. Into the important session where the important impurities like genotoxic impurities, like ICH M7 risk assessment, where all the genotoxic impurities need to be controlled as per the ICH M7 guideline, where the limit of the impurity should be 1.5 microgram, that is a threshold of toxicology concern. So all the impurities, whether it is a process degradation impurities, whenever you are controlling in intermediate stages or in the API stages, need to be checked for the genotoxic assessment. Like, so all the impurities are subjected for in silico studies like QSAR models, that is direct SARA, and to prove that there is no active, like they are not genotoxic, then you can control them as normal impurities. When the impurity is genotoxic alert, then you have to control them as per the ICH M7 guideline. So whenever you are having the specification, whenever you are fixing the specification, all the impurities you are covering that need to be subjected for verification, QSAR verification, where after that, whenever the aims this QSAR positive is there, so obviously you have to control them as per the ICH M7 guideline. If you are unable to control, you have to go for the aims. Then you have to prove that that impurity is not immunogenic and can be treated as normal impurities. And some of the cases, the drug substances or drug product is used for less than lifetime. So LTL approach can be used to have the specification limit for the genotoxic impurities. So whatever the 1.5 micrograms limit is there, that is usage of the drug for lifelong time. Whenever you are using for less than lifetime time, so you can have the approach and you can give the justification to the regulatory authorities and have the you can have the relaxed limit for the genotoxic impurities when the drug is for less than lifetime. And how you will what is the control strategy for the genotoxic impurities in the drug substances or drug product, especially in the drug substances? The genotoxic impurity shall be controlled in the API with the TTC limit. So that is one approach where if the impurity is forming in the N-1 stage or unable to control the impurity in any of the stages, this will be the approach where the genotoxic impurity will be controlled in the drug substances. And the other approach will be control the impurity in the intermediate with the required specification limit. So obviously, with the TTC limit, you have to control the impurity in the intermediate stages. Or we can control that impurity in the intermediate with some limit. Afterwards, we can use purge factor or purge approach, purge studies to control the impurity in the final. So we can have a relaxed limit in the intermediate and conduct the purge studies and prove that this impurity will be complete purged during your manufacturing controls. Then by that, you can have the relaxed limit for these impurities in the intermediate stage. And otherwise, you can use the purge factor approach also for these genotoxic impurities, where you can control the impurities, these impurities at some stage, maybe in the KSM or intermediate stages, and you can calculate the purge factor uh, and show that these impurities will be completely washed out in your manufacturing process. By that, you can avoid the control of these impurities in your drug substance. Obviously, in one of the case where these nitrosamine are covered under ICH-M7 as a class of coat of concern, so they are highly carcinogenic, and because of that, from last 7-8 years, we are facing, all the pharmaceutical industries are facing the difficulties in controlling the nitrosamine impurities. Initially, when the alert came in 2017-18, okay, so it came only because of the small nitrosamine impurities like NDMA, NDEA, and all. But afterwards, the issue came with respect to the NDSRIs and all the pharmaceutical industries now familiarized how to control these impurities in the drug substances and drug product. Now, just I'll brief about these nitrosamines. See, nitrosamines, as you know, okay, so this is having a nitrogen group on amine function. Because of that, it is called a nitrosamine. Then, why these nitrosamines are mutagenic? Okay, so what makes this as a mutagenic? Generally, in the case of genotoxic impurities, as these compounds, the genotoxic impurities compounds are having reactive functionalities. Because of that, they will react with your DNA system in the biological system and form the adducts. But if you see the nitrosamine, so nothing is reactive. So there is only two alkyl groups flanked with amine and the nitroso group. So there is no reactive site in the nitrosamine as such. But whenever you will this nitrosamine enters into biological system, the cytochrome 450 will abstract a proton and forms the hydroxy adduct with the alpha hydrogen of the methyl group or carbon group. So this makes the nitrosamine to form a diazo compound. So the alkyl diazo compounds are highly reactive than any alkyl halides or anything. Because of that, nitrosamines are highly potent, or they are mutagenic or genotoxic. Because of this nature, control of these nitrosamines in the drug substance or drug product is mandatory, and there are certain limits proposed by the authorities, acceptable intake limits by FDA and EMA. So initially we have faced the difficulties in having the AI limits for most of the substrates. Now EMA or FDA, EMA followed by FDA has given the guideline like a CDC approach, where carcinogenicity potential carcinogen approach. So where you can estimate the acceptable intake limit of any of the substance, and accordingly you can control. So like before this guideline, EMA guideline, like 2018-19, so we were in blank in controlling of any nitrosamine impurities in the drug substance or drug product. So whether we have to control with 19 nanograms, 26 nanograms, or 96, so no clarity on the limits, acceptable intake limits of the nitrosamine impurity. Now, there is a defined nitrosamine impurity for some of the nitrosamines in the guideline of the FDA or EMA. Otherwise, if the AI limit is not available, you can approach carcinogenicity potency calculation approach, that is CPCA approach, in calculating the acceptable intake limit and control in drug substance or drug product. And control strategy in most of the cases, all these nitrosamines. Okay, so generally people will control in the drug substance or drug product, because the limit of these impurity is very less in the ppm or ppb level as per the dosages. So controlling in the drug substance or drug product will be appropriate. And whenever NDSRI is there, so NDSRI will be formed when the secondary amine or tertiary amine is there in your API. So that there, okay, controlling of nitrites or peroxides in the excipients also critical in the formulation of drug product. So and the control strategy. So we have to establish the control strategy, how we are controlling these nitrosamine impurities in the drug product or drug substances. So accordingly, we'll put the appropriate limits and proceed with the deliveries. Now, what are the control strategies for regular organic impurities? or residual solvents or inorganic impurities, genotoxic impurities, or the nitrosamine impurities. If you will come to the organic impurities, so high purity starting material. Utilizing high grade raw materials minimizes the risk of impurity transferring into the final product. Okay, so nowadays the regulatory authorities are asking to have the control of the all the impurities, all the unknown impurity at 0.10 level in the starting material. So if the any impurity is specified and unknown, making it known is always advantageous, where you can have the appropriate control strategy for that impurity. So you can have the any limit for a known impurities if you are having appropriate limit. If a unknown impurity is there at a higher levels, other than, higher than the unknown impurity level, then there is always ask the question about that impurities, and at that time you have to have the you have to face the difficulties in identifying the impurity and establishing the control strategies. Instead, while developing the product, identifying all the impurities and having the appropriate controls will be beneficial for the product life cycle. Next, optimize synthesis process. Developing and employing efficient synthesis methods and tight controls over reaction conditions helps minimize the formation of unwanted byproducts. Next is purification techniques. So the purification or the crystallization technique should be appropriate to purge out all the impurities in your intermediates as well as the drug substances or drug product. Next, control of residual solvents. So like we'll use generally class 2 or class 3 solvents in the manufacturing process. So effectively controlling the residual solvents in the drug substances or drug product is critical. Testing and analysis throughout the manufacturing process, stringent quality control measures, including chromatographic techniques and spectrography, are used to identify, quantify the impurities. So here, so whenever we are controlling the impurities in the starting material, the carryover of the impurities in the simultaneous stages need to be done. So obviously, whenever the specified known impurities are there, you have to cut off that specific impurity and the corresponding impurity in the subsequent stages at the unknown level, unknown level of the ICH guideline. By that, so the control strategy will be effective, and we can have less queries from the regulatory authorities when we will file DMF or ANDA. And setting the impurity limits. Regulatory bodies establish acceptable limits for various types of impurities based on the potential toxicity and impact on this. So here, impurity limits. So in starting material or intermediates, you can have the limits as per your trend data. But in your final substance, you can have the impurity limits as per the ICH guidelines. Or if you are having, if the product is non-pharmacopoeial, always you can compare your impurity profile with the RLD and have the level of increased level of the impurity limit for any specific impurity, which is a metabolite or a process related impurity which is not purged out in the manufacturing process. Sometimes the innovator will do toxicology studies for that impurity, will be having higher limits, so that, so by bringing or by comparing your impurity profile with the RLE, you can have the flexibility to have the relaxed level for any impurity in the drug substances or drug product. So these are the general guidelines where all you people know the impurities, organic impurities will be controlled in the drug substances or drug product as per the Q3A, Q3B, residual solvents as per Q3C, and the Q3D element analysis where we'll do appropriate risk assessment and for genotoxic and nitrosamine impurities, you will follow the guideline and control the impurities as per the acceptable intake limits. Just I will give the example, the control strategies in one of the products where we have filed this product for US agency. Uh, Say where this diethyl dienpropyl is a KSM for divalproxypodium. This is a drug where the maximum daily dosage of divalproxypodium is 2 grams. So, whether a known or unknown impurity shall be controlled in the API with a limit of 0.05 only, because the limit for unknown or known is 1 mg in total. So, whether it is a known or unknown, so in the drug substance, you have to control with the limit of 0.05 only. So, if you see the process, so the diethyl malonate derivative is a KSM. Afterwards, only hydrolysis and decarboxylation is there to deliver the API. In this case, the chemical conversions are very less. The regulatory authorities has asked, requested to have a stringent specification in the KSM. Now, we have filed a specification, or we have filed we were having the specification for MSUI limit for unknown impurity as a not more than 0.15. And our trend was unknown impurity 1, 0.12, unknown impurity 2, 0.09, unknown impurity 3 was 0.07. Now, so regulatory authorities here as requested, your MDD is 2 grams. So obviously there are fewer number of chemical conversion after the KSM. So control strategy, establishing the control strategy for all the impurities is mandatory, and have the control of these impurities at 0.05% or show the appropriate control in the so control these impurities in the starting material at 0.05 level or show the appropriate control strategy. This was suggested by the regulatory authorities. Now, so when we have contacted the KSM supplier, he is unable to control these impurities at 0.05 percentage level. So here, when we are unable to control these impurities at required level, we have to establish a appropriate control strategy. So then if you want to establish a control strategy, you have to make them know. Then these when the KSM having these impurities as subjected for the GCMSMS analysis, and we made the 3 impurities known and controlled as a specified known impurities with higher levels, and showed the control strategy for these impurities. And so at the purgeability of these impurities in the subsequent stages by spiking these impurities at higher levels in the KSM, and established the control strategy. By that, so the authorities, the FDA regulatory authority was convinced with our control strategy, and approved our DMF, where our NDA was approved in US for this product. So here we have not controlled these impurities at 0.05 level where the control was mandatory. However, we have established the control strategy. So anything as per the guideline, anything will be acceptable. Either control or control strategy is mandatory for any of the impurity in the drug substance or drug product. So in conclusion, impurities pharmaceutical, whether organic, inorganic, residual, can be significantly impact the safety, efficacy, and quality of drug products. Understanding the sources of and the effects of these impurities is crucial for developing effective control strategies and ensuring compliance with regulatory standards. Though rigorous controls, quality control, and advanced analytic techniques, pharmaceutical manufacturer can minimize the presence of these impurities, thereby ensuring the production of a safe and effective medicine. So only thing is that we should have appropriate control strategy for the impurities, where your process, manufacturing process, will take care of all the impurities, all the possible impurities, and all these impurities we have to subject it for QSAR studies, where the impurities, if they are this QSAR positive, need to be controlled as per the TTC limit. If they are normal impurities, controlled as per ICH Q3A or Q3B level. And if it is a non-pharmacopoeial product, obviously you can use the RLD as a reference to have a little relaxed limit for any of the impurities or any of the metabolite which is observed in your drug substances. Thank you. Thank you, everyone.
All right. Thank you so much, sir, for that amazing session.
Thank
And
you.
I'm sure now everyone got important inputs on the risk-based approaches to impurity management. Now, we will move on to the Q&A.
Right.
Our
Yes.
first question is on your screen.
Yeah.
So,
So, yeah, here is there any timeline for converting recommended AI to final AI for NDSRA as per the guideline?
guidance.
Okay. So, if the guideline publishes the final acceptable limit, we have to stick to that. Okay. And if you are having a CPCA approach in having acceptable in time limit, so when authority publishes the limit, so immediately you have to, they will give the implementation date for that acceptable intake limit, then you have to go. Suppose for vernicline, the FDA has given like some time in having the relaxed AI limits. Like that, whenever acceptable impurity limits are published in the websites in the regulatory authorities, they will give some time. Accordingly, you have to follow the regulatory authorities' procedures in controlling or in adapting to the acceptable intake limits.
Okay. Thank you so much for that. And now we have the next question that's on the screen. Are you able to view it?
1 minute. Under what circumstances can a generic manufacturer rely on innovator toxicology data to establish acceptable intake limits for nitrosamines or genotoxic
Right. So for
impurities, and what are the
genotoxic
scientific
impurities.
and regulatory here? So although the innovator will do the scientific approach, he won't to disclose anything. Obviously, we have to follow our scientific approaches in establishing the limits for nitrosamine or genotoxic impurities. We can't rely. Okay, so whenever he openly publishes, then only we can have, when the innovator can publish openly, when the API comes as a pharmacopoeial product. Until and unless any innovator won't publish his scientific data in the common platform. So obviously, if you want to have a relaxed limit for any genotoxic or nitrosamine impurity, you should have your own scientific data. Otherwise, you can have from regular websites, where if if the structure is already known, then you can use that data, or some lead alerts are there. You can request the authorities to have the limits as per the lead structure.
Thank you so much. So now we have the next question from Parminder Singh, Lotus Pharma. It's, does US FDA and EMA accept finished product or API impurity specification limit based on RLD impurity levels observed, shall request for guideline document on the same.
See, 1 minute. Here, there is no guideline. See, even authorities can't publish any guideline for this type of thing, because they are giving the relaxation based on the criticalities of the product. So sometimes based on the toxicology studies for the impurities and the criticalities which are shown by the innovator, the FDA convinces, when convinces, then only they will give the relaxed limit. Otherwise, they will instruct the companies to follow the ICH guideline to have the specifications. In most of the cases, they won't give the relaxation. Whenever there is a criticality with respect to the process, with respect to the purgeability, or there is a metabolite or a process degradation impurity, then they will give some relaxations for the impurity limit. Suppose I'll say a complex molecule where sugamadex sodium, where the molecule is highly complex and having many chiral centers and all, and purgeability of the impurities is critical. Because of that, although the impurity limit for known or unknown should be 0.1, 0.09, the authorities has given 0.2 or 0.3 limits for some of the impurities based on the innovator toxicology data. And even you can't directly go ahead and fix the limit as per the innovator. You can use that as a reference, and you have to again have to conduct some toxicology data to have that relaxed specification limit. Without any toxicology data, just based on RLD, no regulatory authority will give the acceptance for the higher level of impurity levels in your drug substances or drug product. Yeah.
Oh,
So,
thank you so
if,
much.
yeah, yeah. This is a very interesting question where Fed and Purge approach. Okay. So this is a very important approach where without doing any experiment. Okay. So you can use Purge factor approach. If you want to do any experimental studies, you have to do purge approach. But without purge approach, you can apply purge factor approach, and the given the detailed guideline or detailed paper is published by Tresdale in OPRD, Organic Process and Research Development paper, where what are the factors need to be considered for the purge factor approach. Accordingly, we can justify to the authorities. Most of the times, yeah, FDA or the all EMA, all the regulatory and authorities will convince if your approach is scientifically adequate. So whenever we are calculating purge factor approach, a single person should not do. It should be a cross-functional team approach, where as a team, scientifically, if you do purge factor approach, definitely the regulatory authorities will accept and approve your approach.
Okay, thanks a lot for answering that. We'll now move on to the next question, and that is from Dharmendra Triwari from Medley Pharmaceuticals. What are the key
Yeah,
control
yes,
points in the synthesis process to prevent NDSRI formation?
yes. So, what are the key control points in the synthesis process to prevent NDSRI formation? Now, in the API, for API, so this is in two perspective. First is API perspective. In API, so the reagents or the process should not have any nitrating reagent when NDSRI possibility is there. When nitrating agent is available in the process, it will react with your secondary amine or tertiary amine, which is present in the drug substance, which lead to the formation of NDSRI. If you see the formulation perspective, the excipients or the water, whatever has been used in the manufacturing process of finished product, should have minimum level of nitrites and nitrates, so that you can control effectively the NDSRI. Only here, one, see, for any nitrosamine formation, you should have two components. One is a secondary amine, or the tertiary amine, and another is the nitrating agent. Anyhow, one is available in your drug substance, that is a secondary amine or tertiary amine, that you can't avoid. Only you can avoid is by having controls on the nitrating agent, that is nitrites or nitrating agents from the water or excipients in the drug product or in the drug substances, like any reagents or solvents which is having lesser content of nitrating agents, can inhibit the formation of NDSRI.
Okay, amazing, sir. Uh, we have one more question over here, and it is from Srinivasa Aguram M, General Manager,
Yeah,
Headquarters,
yeah,
India.
right. At what extent the toxicology data is required for generic manufacturers to establish AI for genotoxic impurities or nitrosamine impurities? Now,
Correct.
see, for as a generic manufacturer, okay, when acceptable intake limits are not published in the this one in the regulatory websites like FDA or EMA, you have to do calculation as per the CPCA approach, or you have to go ahead with the lead structure approach and propose the limit. Okay, generic companies to establish the AI limit, the activity is very large, and okay, for a generic player it is not possible. So when AI limit is not disclosed in any of the websites from the regulatory authorities like FDA or EMA, you have to have a CPCA approach to calculate the AI, and accordingly you control. Otherwise, if you are having some a lead structure where the limit is little higher, you can propose that limit by giving that reference to the regulatory authorities that my structure is similar to a lease structure where the limit is little higher. I want to have that limit. At that time, regulatory authorities may be convinced with your literature approach and have the relaxed AI limits.
Okay, wonderful. Now we have a question which from Zia. It mentions appropriate planning and risk assessment help control impurities. Can this assessment be fully done on data-driven outcome softwares? What is the success rate?
No, softwares may not give the appropriate data, complete data. Okay, some extent they will help. Okay, so you have to use as a tool, that as a tool to get an additional data, but you should not depend on only software to have that data. Okay, your brainstorming session along with that software data will be more effective in having appropriate control strategy. Only you should not depend upon the scientific or the data.
Okay, thank you once again for that amazing session, Dr. Praveen. And I'm sure now everyone has got all their questions answered. And now it's time for a well deserved break because we had a lot of sessions piled up after this also. And right now it's 12:30, so we will be back at 1:30 at the scheduled time. All right, we will make sure that we are back on time so that we go ahead with the other sessions which are scheduled for today.
Okay, thank you everyone.
Thank you. Thank you very much, Praveen. So there might be a possibility, just like last time, that we might send you some questions on email as well. because people are mostly sometimes slow typer, and the questions were coming in very slow. And the questions, some of them, they come to my team because we are taking some questions anonymously as well. Those people
Right.
who do not want to share their name, we are submitting it
Yes. Yes.
anonymously for them,
Definitely.
right?
Definitely.
So,
We will ensure to answer all the things. Thank you. Thank you so much.
uh, thank you very much.